So good morning, everyone. It's really great that we have some audience this morning after this wonderful evening. So people are probably still, enjoying last night. But for those that are here, very welcome. Okay, I will talk a little bit louder then. And I think we have a very nice sessions today. We will try to make it a little bit better than yesterday. So we start now sharp at 8:30. I would like to introduce the panel. So perhaps it's nice if you introduce yourself, starting with Rick.
Hi, Rick Avila, computer scientist from Acumetra.
Mary Ellen Geiger from University of Chicago. I'm a medical physicist doing AI and medical imaging and also contact PI for Medrick.
Kyle Myers, good morning, former FDA research scientist and research director, and now regulatory consultant and research collaborator.
Good morning, my name is Anna Walstra and I work for the Institute for Diagnostic Accuracy, where we work on the Forne Lange trial, and I work on the radiological workflow and have a background in technical medicine.
Yeah, good morning, everyone. My name is Mario Silva. I'm a thoracic radiologist from Parma, Italy. I'm involved in the Forne Lange reading process, and I was formerly involved in Italian lung cancer screening trials. Also, actually, currently doing something.
So I think we have a very well-equipped panel to meet with this challenging session that we will have on the CT acquisition parameters. And we had already yesterday, I think, a very nice flavor of what can go wrong everywhere and how it's important to drive this forward. So I think in this session, we will see a lot of problems that are, but also challenges to solve for the coming era. And we would like to see if we have any impact on your opinion during this session. So we will start with some questions for you to answer. And we will repeat the questions at the end of the session. And if there is no impact of the whole session on your opinion, then we have to rethink what we are doing. So please start with the questions.
We don't get to participate.
You will log in automatically. Do you all have the code? Everyone set? Let's start with the questions. Now, the first is, do you think phantom standardization and calibration in lung cancer screening is mandatory? It's a very leading question. And you have several options, as you see here. Only before starting a program, every month, every year, all of the above, not necessary. Okay. That's interesting. You have to understand a little bit about the systems themselves to give the right answer. Okay, it's finished. Then we go to the next question. On the topic of acquisition parameters and those for chest CT screening, do you believe that there should be a standardized minimum, maximum, and average dose? Yes, all of these. Only a maximum dose, only an average dose, not necessary, unsure. That's interesting. Okay, so not a clear answer, but the majority says everything. So a maximum dose, an average dose, and all three. Okay, then we go to the next. What do you think is most important to standardize in lung cancer screening? And you have 4 options: reconstruction and kernels, minimum, maximum, average dose, use of I-ELCAP in detection and segmentation of lung nodules, nodule segmentation algorithms. So it does not give all of those. So you have to make a choice. So that makes it very interesting. Of course, the dose itself has a lot of influence on the outcome of the other parameters. So that makes it a little bit difficult to have a preference there. So there is a preference for the recons and the kernels. Interesting. But also the nodule segmentation algorithms. I personally would have thought that there would have been a different opinion here, but okay. We go to the last one, I think. Should real-time performance monitoring of key parameters of lung cancer screening be implemented at the participant level in lung cancer screening? Yes or no, or unsure. So you could give yourself an opportunity to form your opinion during the session and answer then at the end. So you say unsure here, and then you say yes or no. But if you are really think you know it all already, you can say yes or no. And then we can try to shift your opinion. But this is very nice. So probably we already have a selected opinion here. So the people that are interested in the subject understand that real-time performance, that's what I think is important. So I think this is very nice to start the discussion. I would like to give the floor now to my co-chair, Mario Silva. And first, Anna Walstra will give a short introduction on those key problems that we meet in what we discussed also here with the questions. Okay, Anna? Yep.
But as a first comment, I think it's the answers you've already given. I think it already raises some topic for discussion, especially the first one. And I think Anna is going to have nice case scenarios for that matter. And I think also our panelists are burning with their comments about that, because indeed that was quite a selective choice, wasn't it? And I think the selection reflects what actually we were trying to discuss today.
Yeah, so in the questions, you saw a few topics that we are going to discuss now in the panel. And I want to show you a few results and examples that we've seen recently in the Forne-Langwan trial. And of course, I'll start with the most recent numbers that we've got from the dose levels from the Forne-Langwan trial, because from the 4th of December, we get the dose report from each individual participant is registered in our data management system. And when we had to present this to the EU review meeting, we saw that we scanned with a mean dose of below 1 millisievert of 0.93, which was a very good result. And we'll also be talking about if we can perform one-run low-dose CT in lung cancer screening. We are actually already doing this in the Forne-Langwan trial, where we use a 3rd-generation dual-source CT. And we can use this to scan, we do it for lung cancer, of course, but we also already applied for calcium scoring, and emphysema scoring is now under research. And this is the scan protocol we are currently using, and we are scanning with a very high pitch, which make with no beta blockers, no ECG triggering, and no contrast. And we can still get an accurate calcium scoring, which we investigated by looking at the baseline calcium score and the follow-up calcium score. And we see that when we compare this, there's a very similar score. And these are the two example cases of how we see this in our software, and where the segmentation is a very good match. Of course, there are also a few cases where we don't see a good match. And there were very few cases, but when we went to these cases, we investigated it, and we saw mostly that this was not due to acquisition failure, but mostly due to AI identification errors. So we could see the calcium, but the AI identified it wrongly, but actually the acquisition was good enough to perform the calcium scoring. And then we'll also talk about nodule segmentation algorithms. And we saw that a high number of people thought it was very important to standardize it. And I think this is a good example where we see baseline nodule, 3-month follow-up nodule, and at annual scan, we use a solid segmentation, a solid with manual adjustment, and a part solid segmentation. And you can see there's a large variation here. You can see that we can get a negative, indeterminate, or positive result with these 3 different nodule segmentation algorithms. And especially when we look at the part solid segmentation, this which only focuses on the solid component within the segmentation, and we see there's a stable VDT from 340 days. So this shows there's exponential growth, while the other 2 segmentation algorithms don't show this. We've seen this in multiple cases through our research. And of course, not only within one system, the algorithm can vary, but also between vendors, can be a large difference in segmentation. And this is one case where a nodule by one software is segmented as 90 square meter, and the other one is 103. So this is a borderline case, but it can highlight how it can be very difficult to make sometimes the decision between a negative classification or an indeterminate classification, because there are so many factors that can make this difference. And also very important, reconstruction and kernel standardization. This is an example between a B80F kernel and a B45F kernel, and we see that the solid segmentation is quite similar. When we look at the part solid segmentation, the solid component actually varies with 50%, which is also a very high level of variance, and of course difficult to then make the decision what to do as follow-up for this nodule. Another example from the kernels is that we also compared the kernels, and when we used the iterative reconstruction kernel, it was actually classified as a calcified nodule, making it a negative result and leading to a false negative classification, while actually the nodule was not calcified. And I will also talk about how we want to use our standardized AI cuts use in lung cancer screening. And one of the topics we're investigating also within the For in the Long Run trials, the performance between these 2, and then with the focus on false negatives. And when we look at false negatives, which we here define as nodules above 100 square cubic millimeters at baseline, that were missed by AI radiologists, we actually see that 410 false negatives were due to radiologists and only 33 by AI. And then we followed these up. We saw that 3 of the AI false negatives led to a referral case, while 15 of the radiologists led to referral cases that were otherwise missed. But I think this mostly highlights also that when AI and radiologists are combined, we actually have hardly any false negatives. And until now, we've only found 2 false negative cases. So these are the topics we're going to discuss, and I'll give it back to Mario.
So thank you, Anna, for introducing the topics of this session. And I think you gave an excellent overview of the reasons why we have the title of the session today, Standardization, both for acquisition of data and reporting, because at the end of the day, that refers to the outcome of the test that we are administering to the population. Starting from that, basically the dose level would be the starting point, because that's the acquisition background, the basis where we start from. And that would, of course, determine some changes in the quality of the data. I think most of the people in the whole year are performing lung cancer screening today with low dose CT, probably not all of them with softwares, but we need that. And this paradigm is now the shift that we need to do, to go from visual reading to standardized reading. And I think today it's an excellent moment to discuss about it. So I would like to invite the panelists to give their opinion about dose consideration on low dose CT. Do you think we really need thresholds for average or upper or lower threshold for low dose CT radiation dose. Kyle, what's your opinion for that?
We certainly think we need, I think the easy question is, do we need a threshold on the upper amount of dose? I think that's long been understood. And so the question has been, how low can we push it without having it be that we compromise the diagnostic quality of the image? So I think that's the tougher question, is how low can we go? And that's a challenge to answer that question, because the technology has been evolving in a good way. And the answer today is going to be really different from the answer tomorrow with photon counting CT. And there's a lot still that we haven't figured out yet about how much information to get out of that technology.
Yeah, so I'd say I agree that, so the topic is, we know that too much radiation exposure is bad. So we do really want an upper threshold probably, but we do not really know what the technology can bring to us tomorrow, you said. Yeah, and in the next year. So probably we don't go over that threshold, but we probably don't need to have a lower boundary. So what's your opinion, Rick, on this?
There's real serious issues down at the lower end. On some scanners, if you lower dose aggressively, you give up resolution, especially out in the periphery. So you can actually do more damage by lowering the dose, because you wind up making it very difficult to measure a small lung nodule. So, and it's, I don't believe there's any resource to know which scanners, which protocols, under what conditions. So the only way I know of to figure this out is to put a phantom in the scanner and run a test.
Yeah, and that's exactly the topic, because the thresholds that we're talking about are really near to the variation of our tool. So that's somehow unfortunate. On the other hand, it's fortunate, because we really detect small nodules, but we got to have a balance. And I'd like to hear about Mary Ellen. What's her opinion on this topic?
Well, it comes down to signal detection theory, where you have, and actually that was topic PhD dissertation, so we're going back decades, but it's going to be a combination of the size of the lesion and the contrast. And if you have too low dose, you're going to lose being able to perceive that nodule, especially if it's very small, but even large ones, you will lose it if you have too much noise. So, and I think the signal detection theory is what Rick puts into his phantom calculations.
So I'd like to push it a bit further, because so we're now talking about dose and noise on a baseline. So it introduces an error. And let's make this error systematic over the time, because we know lung cancer screening is about first scan, but is mostly about follow-up scans. Do you think we can accept some error in the baseline if it is then taken over the time, and it becomes a consistent error, a systematic or error over the time?
Yeah, well, yes, I think eventually we could do that, because even with, if you think of deep learning, where you're not just looking at case by case with temporal type analyses, where each one has some variation allowed. There's multiple algorithms out there that can do that, but we have to train it and figure out what kind of variation, what kind of fluctuation at each one can we accept, to say, I have these 5 scans, I can now follow them. And even though this variation is here and it went up, well, that's probably not an increase. It was within that fluctuations. And with enough temporal data, you can develop that. Yeah.
So yeah.
Long, tall, short, wait, long, short neural network, it's called.
And I think we have to understand the clinical relevance of such, because indeed we are talking about nodules, but not just nodules. It's actually, we're talking about early diagnosis of lung diseases here and cardiovascular disease here. Probably that matter becomes even more important when we scan and try to quantify bronchial disease and even more subtle parenchymal disease. That kind of noise can really make a huge difference. And talking about this, one of the one of the topics of the discussion was also about having different reconstructions or even different acquisitions with, yeah.
To the dose issue. So to come up with real advices from your point, what you think that is a minimal dose and what is the maximum dose in a screening application. So yeah, you know that are probably worldwide limitations, guidelines that really said, okay, the mean dose should not go higher than that. For example, in the Netherlands, we had to commit to a mean dose of 1.5 millisievert for the whole screen. So we can prove that we are beyond it, also in the combination with the coronary score. But in other countries, you have a higher dose. The background dose is 3.5 in most Western countries. So on the other hand, we have the problem of the noise itself. So there should also be a lower threshold, quality-wise. So probably could you elaborate on this, starting with Rick and then all of you?
I don't know.
I think what you described as an average dose, hitting a target of an average dose sounds like the right path, because each dose consideration is going to depend on the patient. What's the risk of the patient? How much, you're going to balance the risk versus the dose. Potentially, I think you're going to potentially at times be faced with having to use higher doses because you have a larger patient, things like that. So I kind of like what you described as an average dose target.
And I think we have the ability, knowing the knowledge of how to detect it, what are the risks to the patients, what are the variations from aspects of the scanner, to come up with some kind of prediction model or a table saying, for this situation, this patient, what would be the lowest dose? But there's no one single answer, I don't think.
I like your answer, Muriel. And I was thinking about, this is a personalized medicine question, but it could be bucketed to be able to be something that gives guidelines that are useful and implementable, that aren't on a per patient basis, but on a basis that is relevant for groups of patients that can be considered to be somewhat similar in terms of size and disease manifestation that would be expected. Mm-hmm.
Yeah, I agree. It's very important for the average dose and to also use that and then tailor it to the individual patient. And then for the minimum dose, I don't think you can actually give a minimum dose because it's really dependent on the CT system you're scanning with. Like if we use the photon counting CT, it's of course you get very good image quality at a low dose. And if you scan with a much older CT system, if we want a similar image quality, we get a higher dose. So it's all about setting the minimum or image quality that's needed, and then match the dose to that, and then make sure the average dose is still correct and it doesn't go over the maximum dose.
So you understand that there's a kind of conflict with the ALARA principle. Mm-hmm. So the ALARA principle in the interpretation that many makes it the lower the better. You would not support the lower the better.
Only if the lower the better is with good enough image quality.
Yeah, but yeah. So yeah. I'm still asking you for the minimal threshold as your opinion wise. So not
I can't give a number.
Not evidence wise. So we know now with this we are really looking for the ultra low dose path. You know, with photon counting we could probably come to 0.6, but that's about it. There is some threshold there. I think also for photon counting. So just you are very knowledgeable in this. Where is your feeling that would be the minimal dose? So Rick, again, come up with a figure.
Minimum. Okay. All right. So, okay. So I mean, we're all familiar with the 1.5 millisievert target, right? But on the minimum side, I think noise is going to drive the equation primarily, and I believe that that's going to start happening depending on machine, of course, but down at 0.7, for example.
Okay. I truly think I cannot give a single number because of all the variations involved. So I'll just give 0.7 to go with Rick because I really, you can't give a single number.
Right, right.
Here you go.
Yeah, I think it's just so challenging because there's this natural stress between wanting to minimize dose and wanting access for patients that are only able to be scanned by older machines. And we're not ready to say that the benefit of scanning on an old machine, is not something that we're going to welcome today while we're looking to improve access to better equipment tomorrow. And so, FDA has long said the benefits outweigh the risks for systems that were available in the early 2000s. And now we're just talking about how much better can we do with better technology, but we're not going to take those old systems away.
I think we have a comment from Dr. Kinkelevich.
Yes. It's something that Rick had said, I don't think he expanded on it. That some vendors, when they lower the dose, they still make the images look nice.
Oh.
It's a little secret.
Mhm.
They give up resolution, so you don't appreciate the noise
Mhm.
as much, but you're giving up
Like
resolution
smoothing.
at the expense of dose, and it's, so I need to expand on how much that affects.
Yeah, we've seen some scanners out in the periphery give up 200 to 300% of your resolution, basically by aggressively lowering dose. So sites need to know if they really try and bring that dose down to 0.7, 0.6, 0.5, that their measurement accuracy is going to balloon out. It's going to get much worse.
And it's counterintuitive because dose reduction by itself doesn't change the hardware and reduce dose. It's the fact that they're doing smoothing. And we're going to see more of that with AI if we're not aware and careful in making measurements on an ongoing basis.
And what's good for the human eye may not be what's good for the computer eye. So that smoothing might be done for the human eye.
I think that there's a regulatory side to this or legal side to it. They have to have scanners that with a given dose achieve a certain level of noise. So they're bound both on, well, I have a slide on this, but they're bound on noise and radiation dose, but they're not bound on resolution. So what is the obvious thing to do? Because it's a fundamental trade-off. You just give up resolution.
Yeah, we have several parameters that we can actually tune for reducing dose. And we've been seeing more and more of that. We can act at the level of the tube with current modulation. It can cause some headache, but also we can filter the radiation beam. And we've seen more than one kind of filter for that. Then we can act on the kernel for reconstruction and we lower the resolution. We make images slices thicker sometimes, which is something that we don't want to have. Or we can use then at the end of the day, AI to elaborate everything. So we're seeing a list of options to reduce the dose. And I'd like to ask the panel, is there one solution that you would bet on to say this one could be more standardized over the vendors? Because for instance, the example of the filtering of the radiation beam, we're seeing more filters. So what about the distortion of the signal with one filter or the other filter? Should we start again with Rick from left to right?
Well, so I happen to know that Mario is a world expert on using tin filters on Siemens scanners. And you've gotten really tremendous results with that. So that seems like a really good path, but I'm not familiar enough with the other filters that are out there to comment.
So a small information. We now have also Silva filter from the other vendor. So you mentioned Siemens before. The other vendor would be Canon using the Silva filter. claiming similar results, probably even lower dose. And they also apply already AI reconstructions. But yeah, probably the eye result is pleasant, but as we were mentioning before, from the question from Dr. Yankelevich, the quantitative result is actually something different. And if we move with different filters, then the information and the distortion of the signal can vary. So from the very basic of the signal. So probably I'm guessing the filtering might not be the appropriate way for standardization, because we kind of alter the.
We should say what the filtering's doing, right?
Yeah.
You're taking a polychromatic beam and you're putting a filter in between the X-ray and the detector. I'm sorry, between the source and the detector, and you're basically filtering out some of the spectrum. You're assuming that part of the spectrum is not important to the measurements and the image. But again, I would argue the only way to absolutely know is to put calibration phantoms in the scanner and measure it and validate that you actually have something that isn't really hurting you in the end.
Yeah.
Yeah, I'm going to agree again with Rick. But I want to point out that, we can talk all day on this, and the different CT manufacturers are going to have different models, and across those models, the only way to really know is by putting the phantom in, because they're not going to tell you how they do it. And they're all, the processes and the reconstruction are all proprietary. So you can guess and you can kind of estimate. So we can't go the more analytical route because we're not given that data. So we have to go the experimental route, which are the phantoms. Okay, so we have to answer a lot of questions about what to do in various cases. And we put together this chart a long time ago. If you know the precision of your scanner, you can say something about how much follow-up time you should have before you should do another scan. And if your time window is too short, if you go less than 90 days, you can see that if you have different levels of growth, you're going to have less and less opportunity to see a change, right? There's going to be overlap between the green and the red and the yellow bands. Now, how would you know the precision of your machine? If you really want to get to precision medicine here and say what to do, how would you know that? Well, of course, it's what we've been talking about for a while now. Put a phantom in the scanner, go get the fundamental properties of the acquisition system, which I described yesterday. And you can essentially model what would happen under various different conditions. So what you're seeing here is I went and I took different models of different scanners, including ones that haven't even been built yet. On the left is a 1 millimeter, is a 0.1 millimeter resolution scanner, which we can't build yet. But I put two algorithms at work here. One is a threshold, just pure threshold, and that's the wireframe you see. And inside the purple, that's a pretty advanced canny edge detector boundary method that will basically find, well, it's one of the better methods in computer vision for finding a boundary. So you can see differences between what they can see and what they can do. But it's with these fundamental properties that we can answer these questions. And I just want to bring you back to back in 2016, 2017, I announced that if you put 3 rolls of Scotch tape on a scanner bed, I would predict for you automatically, if you uploaded the scan, the fundamental properties of the acquisition system and the precision and bias of measurement of small lung nodules. We took all that data, which you can see here from 27 international sites, and we plotted that resolution versus noise. And we got the fundamental property, the trade-off between resolution versus noise. And where do you want to be? You want to be at that elbow. So when we talk about resolution versus noise and how much and dose, which of course impacts this,
it's going to be based on what the physics will allow. What will the scanner allow, and what can your phantom tell you about those fundamental properties? Also, I want to point out during COVID-19, we pulled together an international group, many of whom are here, to come up with recommendations for how to do acquisitions to study COVID-19 as fast as possible and as high quality as possible. We published this, and inside of it, we gave recommended recon kernels. So now what we're saying is basically we're saying to the vendors, these recon kernels are really important to us. Please don't change them. And I think this is the way to go. I think the community needs to settle on a few recon kernels that they really trust for different clinical applications and stake the claim. Say this, we can't afford to have changes in these over time. Lastly, I just want to say the only way to really answer these questions is a multidisciplinary approach with a real emphasis on first principles and fundamentals. And that's consistent, I think, with what we've been saying here.
Thanks, Rick. Great. May I ask you to get back to your first slide? Because I think we get an incredibly interesting information from your graph here. If you take a look at the time interval to detect with some certainty the actual growth with differentiation of volume doubling time, you more or less get to 150 days, which is the 6-month control of the long grass 3 category for the indeterminate nodule, those that where we want to make some kind of difference. We can debate about 3-month or 6-month follow-up, but I think everybody of us scanning a small nodule after 3 months would be much happier to be at 6 months. And the graph shows the reason why, because we kind of go beyond the noise of our machine and can get much more consistent information over the growth rate, which is at the end of the day what really matters for that single nodule over 500 that will become a cancer. And I think this is a great theoretical demonstration of what a 6-month follow-up means actually in clinical activity for lung cancer screening. Thank you, Rick. Great. Rick is giving all of us many phantoms, and we're very happy to spend some weekends over the scanner. How many hundreds?
One remark here.
Yeah.
6 months can be too long
I know.
in small cancers.
Mm-hmm.
So that should also be taken into account. So for the kind of methodology you would like to do 6 months to get more reliable results, but the interval for small cell lung cancers is then too long. We have some very convincing cases there.
Yeah, but we talk about this every Monday, more or less.
you have control over the width of that green bar. So if you do the right things on that scanner, even an older scanner, you can bring it down.
Yeah. So if you want to catch the small cancers, you need a certain methodology and requirements to your system that gives you not the freedom to do this all.
Yeah.
That's
agreed.
the point. Yeah.
Yeah, and I think compromise to get to that point is to use the kernels that you suggested. But when you read CT images with that kind of kernel, as a radiologist, I would say you're not really happy, because morphology then as you get like a visual quality, which is something different compared to what you're used to. Probably here's this is the moment to get back to Anna's slides when she showed the protocol for reconstructions of images in the long run. You saw 4 reconstructions, 4 columns, and one was for the visual reading, one is for the quantitation of nodules, one would be for the emphysema, and the last one, much thicker, 3 millimeters thickness, 1.5 increment, is for coronary calcium evaluation. So here, I would like to get back to that question. Do you think that one acquisition and multiple reconstructions is okay for imaging both early pulmonary disease and early cardiac disease? Or do we need separate acquisitions to stick with temporal resolutions and other parameters?
Yeah, I can respond to that. Well, I have showed that in the For the Long Run trial, we're already doing this. And we showed that it is depends on the scanner system you're working with, if it's doable. But we showed that you can do trustworthy calcium scoring and lung nodules, and emphysema is now even being researched by one of our PhD students. So I think we clearly showed that it is possible, but it's not possible on all our systems. We have a few centers which scan with all their CT systems, and there we just focus on the lung nodules. So you have to make that comparison and don't try to do it as well if you know the image quality is not good enough.
Can I ask, do you have a sense of what you would tell people as, some sort of boundary between the systems that can and cannot support doing this in a single scan then? It sounds like in a way you've discovered through the back door.
Yeah, we've now doing it only on our dual source CT systems that can scan with a very high pitch to give a temporal resolution that's good enough. And on the other systems, we just focus on the lung nodules. So. But I think in the future, we can also discover new maybe protocols and see and test if it's also reliable. But that's more in the research phase, and we're not reporting that to the patients yet.
So I'd like to just say, come back to this kind of broken record of it. If you go down to the fundamentals, the resolution, not just in spatial domain, but in time. Right, that's what you were referring to. If you know the performance of the scanner in terms of resolution, in terms of noise, and basically the fundamentals that I showed before, you can answer the question I think very clearly whether or not a certain size calcification is going to show up or could get missed. I don't think we have to experimentally test every scanner in the world with lots and lots of different protocols. I think we can pretty much determine with the data we have now what your best bet is in order to do this simultaneously. So. And that's what we're doing right now with this guidance we're putting out for COPD or emphysema and lung nodules. Is we're again starting with first principles and coming up based on the data we have now, what would be a good protocol for many scanners.
Yeah.
Right. And I just wanted to add that if we didn't have all this proprietary issue of not really knowing how the data is being reconstructed, a while back, we went back and we did play with reconstruction in a simulated way with an I-ELCAP phantom, where you could simulate the acquisition, the slices, the patient, the cardiac, the actual calcification, and you could reconstruct then based on the phase. And it might be with the heart beating that if you just started that reconstruction at the wrong point, you missed it. But if you started how many milliseconds later, you wouldn't have blurred it out, that calcifications. And of course, if you had multiple ones, you'd have to reconstruct differently. And the goal was to come up with the AI technique to say, this is blurred, but if you go back and reconstruct, and this is all post-acquisition, you have the data, but if you go back and reconstruct, you can see that nodule better. And I think that kind of high level of AI incorporating, finding something, saying, well, you're not at your best right now, but if we reconstruct starting at a different point, we can now get a better nodule view out, and then assess it. So all these components, acquisition all the way to interpretation, can be put into a bigger model.
Yeah. So phantoms, standardization, we've seen the question in the beginning of the session from Professor Oudkerk. How often should we scan the phantom? And how often should we standardize the CT scanner? And do you think this should be on a personalized and a real-time manner? So scan a small phantom with each and every patient to have a reference and start from there.
So we've been exploring this for a couple, well, at least a decade. We looked at with David and Claudia and others around here. We looked at pocket phantoms. You saw that with the Abigail study data that we showed yesterday. The problem is it's just a little cumbersome to put a phantom on the chest of a patient, do that kind of stuff. So the other avenue we've gone down is a table phantom. So the idea there is that embedded, we've got it down to about 11 millimeters above the original table height position. We can put all the components to measure everything that I showed you before, but it only happens at a certain distance from isocenter, a certain range. And so what we can do with that is that we can validate that the assumptions we're making about the quality of the scan are true based on what we see in that slice across the scanner table, let's say arc. But I think what the exciting thing that's possible though is that on every slice that the CT scanner produces, in the DICOM header, we could be putting the resolution, the noise, all these fundamental properties, which are so important, in the DICOM header. And then all software downstream can leverage that information. And we know that that's useful to do better quantitative measurements and detection. So I personally think we're all headed toward table phantoms, and scanner manufacturers will eventually incorporate those types of technologies into their table.
So regulatory reference table phantom would be, yeah, the way forward to standardize on a real-time personalized manner. Super. I think Mary Ellen has some questions, some slides, right?
Yes.
Staff is preparing it.
Thank you. So my slides, the two to start with, basically have to do with once you have the data acquisition, how do you standardize it so you can use it either for reporting, for AI, for others? And this follows, you all learned about MIDRC yesterday, so I won't go through it since I don't have much time. So for these slides here, basically, within MIDRC, we have a data model, and this allows us to search and build cohorts. You can expand this to clinical work where you're looking for maybe comparison images or how you organize your reporting system. But to do that gets very complicated. So here, for example, is a list of CT images. These are lung CTs. And you'll notice here, these are all the same type of images. But in the DICOM header, everyone, they have different names for them. People can't agree to call a certain type of image one name. So they come to us with different names. And so we're using the LOINC mapping, which came from the Redlex playbook. And basically, we use this to harmonize the name of the image. And now we've harmonized it. As you can see here, they're all CT chests without contrast. Why everyone couldn't say that at the very beginning, we're not sure, but they don't. So when we have images from multiple institutions, harmonize it. So now we can search and go through it. And by doing this, we've taken over 146,000 studied descriptors, these long names, and matched them to just 65 unique CT names. It makes, because if you're going to build a cohort and you have to remember all these different names, it will take you for a long time. And accuracy around 97%. This LOINC mapping, we keep expanding it as we take in more types of images with different types of names. We put it on our public GitHub. Any of you can download it if you want to use it in your own lab to standardize the names in your CT scans, and we're now expanding it. So use it if you like. It's all open and free. And we also have integrated into our overall pipeline so that when someone comes and uses the user portal, they can search, and you can see there's LOINC is an option to find what you're looking for. And this really helps standardize. So this is post-acquisition, but useful in that way. And of course, free and open. We also working with phantoms. I think we could have more discussion on that, but we've had a lot today, so I won't go to it. This is the work of Paul Kinahan's group, part of Midrick. Looking to, when you're trying to do, again, build a cohort and you want a certain quality of images, keep in mind, Midrick has curated data. That doesn't mean high quality data. That means curated, meaning ultimately we want to be able to say, we have this data, it's not that good. We have this data that is good quality data. So curated means you've kind of said what's good and bad, because with AI, if you only train with good stuff and you go out in the real world, you're going to have not so good stuff. So this is kind of, again, a post-acquisition, trying to put a quality label on the images that we have there, so could enable cohort building. I'll stop there. Thank you.
Thank you. It really fits perfectly in what we've just said. And standardization, yeah, is important. One would like to have like just one reference, one vendor. But I think we're lucky that in this slide you show with 4 main vendors, probably the 5th is coming up. It's not too much to standardize on, right? Sometimes you have like 20, 50, hundreds vendors that do one thing, but we're starting with a small crew here. So at some point there's some kind of hope to get it to a standardized data.
Yes.
So and I'd like to continue on this, talking about the availability of the standardized coronary calcification metrics. You mentioned before the reconstructions in different phases of the heartbeat. Yeah, it's probably still quite far from the lung cancer screening acquisition, isn't it?
Well, once you figure it out, it's all done in a computer, and the computers are getting faster. Picture the scenario: the patient has an image, the image comes in, there's something seen by the computer, it's too blurry, you can't quite tell. You say, go back and reconstruct it a few seconds earlier, a percent of the heartbeat earlier. And it reconstructs and shows you, which would enable both improvement in human vision and computer vision. But that feedback loop could be, I would expect, could be built in. And we did this over a decade ago, so I'm sure computers are faster now.
Yeah. So probably the way for the perfect cardiac imaging would be to have a dedicated ECG gated on the cardiac volume.
Right now, low-dose CT for this is not gated, is that correct?
Yeah, this
Right,
is not
but you could
gated, sure.
pretend it is,
Okay.
right? You could say a typical heartbeat is this amount. You could say there's something there I can't. Let's just go back and reconstruct it, that percent of a heartbeat backwards. Even without gating, you can and optimize for that focus nodule to see where, because you've already said there's something there, I just can't quite see what it is. And you can go back a certain couple of percents and still have it come and get an image quality assessment on it, particular to that location within that part of the lung, if it's being that affected by the heartbeat.
So probably one direction would be to lower the pitch when we scan over the heart. Z axis.
Now you're getting out of my area, but yes, I could see that.
So yeah, so probably I think some vendor is already proposing this kind of acquisition. They go non-ACG gated where the ACG is not needed, then over the heart is ECG gated, and then you go back to non-ECG gated. And what you were mentioning is you just need to have the data set. So you pretend that's ECG gated, even if it's not. So you just lower the pitch at that point. So,
but when you say you just need the data set needed is the one prior to reconstruction, which is proprietary and typically not given. But if we have that, we could play this game.
Yeah, that's been an interesting development of the acquisition. Have everybody ever explored a lower pitch over the heart to reconstruct a different time points?
Increasing is already for I think 15 years an issue because you get data gaps if you stretch it too much. So this again a kind of trade-off between temporal resolution and spatial resolution. And yeah, I think that the optimum there is only experimental to be determined. And I think in this sense, Thomas Floor did a fantastic job to get it optimized in the Siemens systems. So the acquisition that we are using has been optimized and designed more or less by Thomas Floor himself. So that's why we are very trusting that this is probably near the optimum. So
What you're saying, you have to keep pushing on the way of higher pitch because the higher the pitch.
that on one hand for the temporal resolution,
Yeah.
but not losing too much data because of the gap. So don't go to the ultimate, but try to find the optimum. And I think the pitch we are using now, yeah, that is the whole designer of this stuff. Also the increase of the table movement, what you want to lower down and then increase has huge impacts on everything what you do. You cannot do it just like that. You need to get a stable movement. You need also 1 second before and 1 second after. You have to go up and slow down. So that is not, and you cannot just in 1 second slow down. It's impossible, or in a fraction of a second. It influences everything you do.
You're
So-
continuously acquiring.
So in my opinion, the whole measurement must be in one speed fluent, and you cannot change the speed in between.
Yeah, I agree. Yeah, I wasn't changing hardware. I was changing software. Yeah, I definitely don't change hardware. But software, you can do it. You can, because it's continuously acquired.
That will really help for the sake of those that we started with. Because as soon as you increase the pitch, that would lower the dose, and you'll be happier as a clinician, probably not as a physicist every time. But
I'm not a clinician.
I think we now could try and see what the answer are now if we take another round for the 4 questions. Here we are again. Like to see whether this changed it, what the discussion that we had gave you some different impression. So the question now again is, do you think that phantom standardization and calibration in lung cancer screening is mandatory? So options are only before starting the lung cancer screening program, every month, every year, anything before, not really necessary.
By the way, we've been pushing people to do it quarterly.
Yeah. Every hour.
Every slide.
Yeah, everything.
That's very wonderful.
I think, yeah, after the discussion, so if I read it correct, now people think that the scanner has to be standardized every year. And I'm quite impressed by the answer, I have to say.
I'm surprised.
I want to stretch it a bit because I think Rick has some information for us for the specific purposes where the standardization has to be repeated every day or every time the scanner moves. What about mobile scanners, Rick?
Yeah. I showed a chart yesterday. I mean, one site was just having tremendous trouble keeping the quality consistent. And we've seen that with some other scanners and some other devices, mobile scanners. Unfortunately, there's things that can happen. Of course, you can have equipment failures, but certainly if you change, what we say is do it every quarter. And if you have a known change to the scanner, for example, you might have a software update, you might have a hardware update, or you have a failure and you have to have a repair. Just just do it to maintain peace of mind. Of course, the big question comes, what happens if you see a big difference? Let's say you get a new scanner, you got it, you do the calibration and you get a much bigger difference. Mario ran into this one weekend a couple of years ago, right?
Yeah. So we changed the scanner and all of a sudden the kernels were different. So we had our perfect lung cancer screening protocol at very low dose. We had a new scanner. Everything was blown up. We have to do it from scratch again. But yeah, it took probably 100 scans and more reconstructions, but finally we got it. We got it.
One weekend I saw like 100 scans come through the phantom analysis system, and I realized it was Mario, and I called him up and I found out what was going on. But
Yeah,
but the
no
good news,
hacker.
the good news is that once you had settled on what was a better protocol, we could then tell everybody else in the world what to do.
Mhm.
Yeah, I think one thing is not clear from the answers, I think, that several people, I think, have the idea that if the system is okay at one point in time,
It'll be.
it will be okay the rest of the
Yeah.
time. So you should all comment on this.
Oh, so I will say these answers are scary. Is that what you want me to say? These answers are scary in the sense that it's too complacent, I think, with, you know, you did it one time that year and it looked good and you stopped doing it. But if, you know, if standardization and calibration can go automatically, which Rick is moving towards, it could be done every time.
Yes.
Mhm. So, but not to just do it once a year and say we're okay.
We have to conclude that there is a major disagreement between the panel and the audience.
We had
So maybe we could hear from the audience on why,
Right.
you know,
Yeah.
yeah, it'd be
You're-
good to understand that a little
Thank
better.
Dr. Moshanas.
This is a phenomenal panel. Thank you so much. But one of the things to people that don't do this every day that should be known is that CT manufacturers are trying to sell their products. They're selling their products in the marketplace today to radiologists, chairmen of Department of Radiology that are looking for basically imaging, an imaging tool. And what we're in this room kind of talking about is moving CT to a measurement tool. And we've heard this described with computer vision versus, you know, visualization. And it's not that anybody's evil. It's just that no one comes for a scanner. I want to buy your latest and greatest to do measurements. It's just not penetrated into the market. And once it does, people will be a lot more attentive to these issues. They'll get solved in the background and you can just, you know, do stuff. But for the time being, visualization is king. And that's okay because for lots of things, visualization is great. And we got the NLST, you know, on visualization. But we can do a lot more and a lot more over time with these annual repeats of scanning and stuff like that. And starting to look at multimodality, quantitation is going to become ever more important. And there's going to be this tension. And if people don't understand this, they don't understand what Mary Ellen said, you know, if you have these curated things, pretty good quality versus great quality, you can have much different performance of AI tools and everything else. So all this stuff that's, you know, published now, when it moves into the real world, there's going to be, you know, different outcomes. This is something we all have to be aware of and kind of live with and try to optimize. And this kind of discussion is perfect for that.
Thank you, Jim. So should we try and see if the second question is? Okay, perfect. So second question again, on the topic of acquisition parameters and dose for chastity screening, do you believe there should be a standardized minimum, maximum, and average dose?
Kind of ambiguous, to be honest.
Mm.
So I could say minimum 0, maximum 0.5. We're done. Average
So-
0.75.
But trying to understand, I think it's interesting, isn't it? Because there was, okay, so there is consistency. I think on the only maximum dose for people are still there. But some of them is changing their mind. Whereas I think there is this unknown part of the bar, which is probably somebody who didn't vote before, is it? Could this be? Who knows? So basically, most of the people remain with their idea, and the majority here is again with all of 3. I think we can see again the reason why we're talking about dose reduction, because that's the biological risk that is determined by the radiation exposure on the one hand, which we know, but on the other hand, on the lower hand, we know that we might be gaining consistency via technology, via AI, and via increased sensitivity of the detector, which is currently the case of a continuously increased consistency of the detector. But we have also heard from the specialist, from the physicist here, that below a certain dose, yeah, but expert, let's say the expert, Rick, below a certain dose, we anyway will have problems with the quality of the data. We always have to balance whether the quality of the data is meant for a 1 millimeter nodule or for a 10 millimeter nodule. I think that's probably a question that still needs to be answered, because yeah, we need stable data until which threshold? Are we asking for a 0.5 millimeter precision in the measurement of the nodule? Are we asking for a stable error along the screening procedure? So we have kind of a stable error there. I think the matter is still quite open for the discussion. And probably a further variable here is that if we pretend that the error is systematic over the screening period, actually we have to be aware that we are going to change scanner.
Yeah.
Probably every 10 years, but we are going to have software updates probably every 6 months. So probably that standard error that we were talking about before is not that controlled. So we have to listen to Rick and Mariellen and Kylie and Anna when they say also a lower limit is required, because this is what we know now. Okay. We can go further with the next question. Question 3 again. What do you think is most important to standardize in lung cancer screening? The reconstruction and kernels, so basically the data that you will be working from that time on. The dose, which somehow sums up with the previous category. The use of AI and computer-aided detection in the detection and segmentation of lung nodules, or finally, on nodule segmentation algorithm. Again, there is quite some consistency, I think, for the reconstruction and kernel, which is the basis. Please, Mariellen.
I always worry when the question asks for the most important, meaning that we not have to worry about anything else. And so I think this can be taken the wrong way. I think here, I would say, I would, if I was forced, I would have to say the first one also, but I would not ignore the others, and I think we need to do this in a multi aspect analysis.
Yeah. Probably I think most of the people acknowledge that there is something which is the basic to work on, which is the data. Then we're now getting used to the variability of the softwares. So probably the software can, I think the back thinking is that the software can always be adjusted, whereas the data is not.
The original data.
Yeah, yeah. So I think we have a last question, right? Should real-time performance monitoring of key parameters of lung cancer screening be implemented at the participant level in lung cancer screening? So is anybody happy with the in-table phantom, basically?
In-table phantom with feedback.
Real time.
Automatic,
Real time.
in real time feedback.
Straight into
yeah.
the .com header.
Mhm.
Yes, that's what it should be.
I think we did a great job. Congratulations, panelists. 88%, 89 is agreeing. Even more. I think this is a success, isn't it? So with this final argument, I'd like to thank all the panelists and all the audience for participating in the session. Our time is now over. Thank you, everybody, for making this session so interesting and engaging, and enjoy the rest of the day. Thank you.
Thank you.